This presentation is by Mary and I, how IRE1alpha drives lung epithelial progenitor dysfunction to establish a niche for pulmonary fibrosis.
Injuries to lung epithelium can usually resolve with minimal residual effects. However, some injuries do trigger excessive extracellular matrix deposition, resulting in pulmonary fibrosis. After lung injury, damage-associated transient progenitors, which we will now refer to as DATPs, derived from airway and alveolar epithelial cells, emerge and expand. Lung injury can be due to things such as bleomycin, epithelial cell ablation from diphtheria, which is a toxin, and viral infections such as COVID-19. These progenitor cells are in a transitional state between the damaged epithelial cells and between newly regenerated alveoli. They express profibrotic genes, which could lead to the promotion of idiopathic pulmonary fibrosis or differentiate into mature alveolar type 1 and type 2 to regenerate new alveoli. So basically, these cells have a dysfunctional phenotype after injury, which limits their ability to differentiate into new alveoli, and instead they express genes leading to pulmonary fibrosis. But the pathways that induce and maintain these DATPs are not fully understood. Researchers show that IRE1alpha is a promoter of DATP abundance as well as function. To do this, they give a nanomolar potent monoselective kinase inhibitor of IRE1alpha, KIRA8, or conditional epithelial IRE1alpha gene knockout to mice, and it did reduce DATP cell number as well as pulmonary fibrosis after lung injury from bleomycin.
In this research, C57BL6 mice or mice chondrogenic to the C57BL6 background were used. To induce fibrosis, 8 to 14 week-old mice were anesthetized with isoflurane or ketamine and xylazine. Then they were exposed to a single dose of intranasal bleomycin. Next, the mice were euthanized to harvest the lungs. KIRA8 was dissolved in 3% ethanol, 7% tween, and 90% normal saline and injected peritoneally at 50 milligrams per kilogram daily starting on the day of bleomycin exposure. Anti-integrin beta-6 antibody, 3G9 or control antibody, was dissolved in normal saline and injected at 10 milligrams per kilogram 2 times a week, also starting on the first day of bleomycin exposure. Additionally, tamoxifen was dissolved in corn oil and administered by gavage at 200 milligrams per kilogram at indicated time points.
In order to determine if IRE1alpha activity was required in the epithelium for pulmonary fibrosis, mice were exposed to bleomycin. Wild-type and epithelial knockout mice showed acute lung injury at day 8. Compared to the wild-type, knockout of IRE1alpha in the epithelium showed that these mice were protected from lung fibrosis based on day 21 lung hydroxyproline content measuring collagen. When there was less collagen, there is less collagen accumulation around DATPs, keeping them in the transitional state. So they are able to leave the transitional state when there is less collagen, which is why these mice are protected due to the fact that there will be less fibrosis. As you can see on the left, saline is the control, the middle is the wild type, and on the right is the knockout.
They then stained histological sections with picrocereus red and captured cross-polarized images. This highlights collagen networks. It was shown that lungs from the knockout mice had less staining. Figures C and D show that IRE1alpha activity in the epithelium is required for lung fibrosis because without it, there is less collagen, and collagen that surrounds DATPs leads to lung fibrosis by causing a buildup of fibrotic tissue, interfering with the functioning of the lungs and preventing DATP cells from leaving the transitional state.
To determine if IRE1alpha regulates the profibrotic phenotype of DATPs, KIRA8 was used. This figure shows RNA in situ hybridization, labeling RNA for KRT7, a cytokeratin, in teal, and ITGB6 in magenta. ITGB6 is involved in TGF-beta-induced gene expression leading to fibrosis because it activates pathways that result in excessive production of extracellular matrix and inhibition of extracellular matrix degradation, followed by washing and restaining with picrocereus red for collagen. Closed arrowheads denote KRT7 and ITGB6 double positive cells, where arrowheads that are outlined indicate KRT7 cells without significant ITGB6 expression. In KIRA8 treated mice, the presence of ITGB6 cells was decreased, which is shown here.
This figure shows that there is a decreased expression of TGF-beta genes in mice treated with KIRA8 inhibiting IRE1alpha or with 3G9, which is an anti-integrin beta-6 antibody. The heat map shows the expression values for TGF-beta induced genes in epithelial ribotag libraries. Epithelial transcriptomes were obtained, and purple shows lower expression, which you see in the control and in both treatment groups, but less purple, and higher expression of red seen in the bleomycin control, where IRE1alpha is present. The expression of TGF-beta genes would lead to fibrosis by modulating fibroblast phenotype and function.
Just to wrap up the results that we saw, the knockout of IRE1alpha resulted in less collagen buildup, leading to less fibrosis, proving that IRE1alpha does play a role in expression of fibrotic genes in DATPs. Also, ITGB6 was decreased when IRE1alpha was inhibited, and TGF-beta induced genes were also decreased when mice were treated with KIRA8 or 3G9, and this makes sense since ITGB6 was decreased. And these results show that IRE1alpha play a big role in promoting the fibrotic phenotype of DATPs, leading to pulmonary fibrosis after injury of lung epithelium.
Thank you for listening, and here are some questions that you can think about. What is bleomycin and how does it damage the lungs? Additionally, being that IRE1alpha persistent signaling enhances TGF-beta transforming growth factor, what disease besides pulmonary fibrosis would IRE1alpha be involved in?